You can see this is a hippocampus signaling pathway.
Yeah.
So how we prepare the network of, for example, when someone is saying PAT-J is overexpressed or downregulated, then it means there will be change in hippo signaling function.
Function. Okay.
So when we are saying there is alteration in gene, overexpression in gene or down regulation of the genes, it means those pathways which are connected with these genes are altered. So do you understand how these proteins and genes are connected with the functions?
Yeah.
So when somebody is saying I'm focusing on these genes, it means they are looking for certain specific pathway component.
Yeah.
All these genes are the components of hippo signaling pathway.
Okay.
Second thing, the one gene is not connected with only one pathway. It may be connected with more than one pathways also.
Okay.
So when the genes are connected with more number of pathways, it means that that gene has, if there is any change in that gene, they have the chance to impact more pathway functions.
Okay.
So more disasters could come up as a result of alteration in those genes.
Genes. Yeah.
Clear?
Yeah.
Now you might have also heard from me that some people also focus on mutations.
Hmm.
So when somebody is saying there is mutation in the particular gene, it means that when there will be mutation in a functional region which has the chance of interaction with other proteins and other components, it means that there will be functional impact also.
Yeah.
And these things you might have heard in case of cancer and many other disease, that this is the particular mutation, because of this these diseases arise. So that means the mutation which take place on particular gene or protein, that mutation may also lead to s- bigger functional changes, alterations.
Hmm.
So is it clear now? Complete idea why everyone is saying that I am focusing on genes and functions, cake pathway, gene ontology. Do you understand now?
Yeah, bro. I understand.
So scientific reason, logical reason, everything is clear now.
Yeah. So I think it was like I mean, pretty basic in term of like it's not basic, but basic in a sense like all the gene like can regulate like down regulate like if you just go back and it can uh, impact on protein synthesis, right? So if there is a one problem in one gene, so
Many things.
whole pathway will not work.
If there is, if there is functional mutation change in the gene,
Mhm.
then protein expression could be affected. First thing.
Mhm.
Second thing is, if there is no change at gene level,
Mhm.
there is change at protein level, the mutation in the
Mhm.
protein,
Mhm.
then there is a direct function changes. But
Okay.
because of gene alteration, there will
Mhm.
be no protein synthesis, relevant protein synthesis, and there will be definitely mm, complete aberration at functional level.
Mhm.
So everything is connected with each other.
Yeah.
Now, third thing comes other than mutation and gene expression, that is epigenetic regulation.
Okay.
These days, there are large number of researchers who are also analyzing the epigenetics data.
Okay.
And one data is called DNA methylation.
Yeah.
Another data is called gypsic data.
Okay.
Third data is called ataxic data.
Okay.
All these three types of data belong to epigenetics data.
Okay.
So, first part is gene expression, second part is mutation, third part comes epigenetics, and
Okay.
epigenetics mainly focuses on gypsic, ataxic DNA
Methylation.
methylation.
Yeah.
Clear? Any question which you could like to ask here?
Not here.
Okay.
Yeah.
So this is also clear now.
Yeah.
Okay. Now, what we discussed currently is, for example, expression data, mutational data, epigenetics data. If we want to prepare the network in all the different analysis part, then we can say that, for example, when we say expression data, then it is differentially expressed genes network.
Mm.
When we say mutational data, network for mutational data, then we can say these are the genes which are mutated lots in our case, and for those genes we prepared the network. Then we can say this is mutational data network. Third thing is epigenetics data network. So these are the way how you can prepare the network. And of course, protein is already there. So it depends when and where you want to, what do you want to prepare. Okay.
Okay.
So this is not any big issues which I you could say, okay, we should do like this, we should do like that. It's all about your independence for which type of data you are preparing the network.
Okay.
Okay, it's
Okay.
very clear. So next thing, so this is like most of the things clear regarding the network
Mm.
concept. Now I'm so now I'm asking some questions.
Yeah.
For your data set, you have already prepared the Excel file.
Yeah.
Yes, Excel file has the gene names, fold changes, event.
P-value.
What you need to do?
Sorry, could you repeat it?
Did you also shortlist it based on plus minus two?
Yes.
Perfect. So what you have to do now is
Yeah.
first step will be list of differentially expressed genes. That is
Mm.
X. Is it clear now?
Mm.
So you have it with you.
Yeah, I already made this.
So that means gene level analysis you have done it.
Mm.
Do you remember that last time I have discussed something with you regarding the analysis step? Do you remember this software, Signable?
Uh, I think we haven't discussed about this one before.
Mhm. Okay.
Okay.
So one thing what I recommend you to do now
Okay.
is I'm stopping sharing this thing,
Hmm.
and you need to paste your genes here
Okay.
in this block, and
Okay.
then just click here submit.
Okay.
So first I'm stopping sharing, then you have to share this